So enzymes are very versatile. They're chemicals which can become denatured. They're non-living, and they help us by facilitating the breakdown of large molecules into smaller ones, or help us by changing molecules around, or also by taking small molecules and making them bigger. So they're very versatile. The good thing about most enzymes is they're quite logically named. As you can see from my title, that often it's the product they work on plus A's added. So proteins, protease, lipids, lipase, starch, unfortunately is amylose, so it's amylase. You've got carbohydrates, carbohydrase. So they're quite logically named. So luckily, generally you can guess what the carbohydrate does from its name because it's a good giveaway. We're also going to look at how the bile works and helps with digestion. So the first thing we're going to look at is glucose, and glucose is the single molecules which make up most sugars. There are others as well, but glucose is the one we're going to look at GCSE. So the sugars themselves, often referred to as carbohydrates, are worked on by carbohydrase, which is found in the mouth and the small intestines, and it breaks down those long chain carbohydrates into simpler, smaller sugars. One of the biggest sugars we know is starch, which, as I said, the correct name for is amylose, and that's broken down by amylase in the mouth to start off with, and also in the small intestine from the pancreas. It's because starch is such a long chain molecule, it needs to be broken down in many places. So we start it in the mouth, it then stops in the stomach due to the pH of the stomach. It's too acidic for amylase to work. And then pancreatic amylase takes over again. So we've got salivary amylase in the mouth, stops because of the pH of the stomach, the acid there, and then pancreatic amylase takes back over and continues to break the starch down into the glucose molecules. Next enzyme we need to look at is protease, and that breaks down proteins by breaking the peptide bond so the amino acids are liberated from the long chain molecule of proteins. It's found in the stomach. It's one of the few enzymes that can work in the low pH of the stomach. The stomach's about pH 2, so it's very acidic, but it is capable of working at that pH. In fact, it denatures when it leaves the stomach. So digestion of proteins occurs mainly in the stomach, but there's also some going on in the small intestine from the pancreas again. So protease breaks down proteins into amino acids. There is a couple of ones that have got their own name, such as pectin and stuff like that, but the broad term is protease. And following the same principle, we have lipids or fats being broken down by lipase, sometimes referred to as lipase, depending if you prefer. I prefer lipase, but that's just personal choice. Fats are again broken down in the small intestine from the pancreas. We can see how important the pancreas is. It produces loads of digestive enzymes. The gastric juices, for example, are the common term for all the digestive enzymes. And what they do is they break the big triglyceride into smaller fatty acids and glycerol. So it breaks apart the three fatty acids. That's why it's a triglyceride, three fatty acids in red, and it leaves behind the glycerol shoulders, the bit in green there, and all those are now small enough to be absorbed from the small intestine into the bloodstream. So for enzymes to work, they need specific conditions, as we saw in the video about that. So we have to keep the conditions as near perfect for the enzymes as possible. And the temperature we keep them at, because our enzymes are adapted to function at our temperature, our optimum is 37 degrees. It only really fluctuates due to illness or due to extremes of temperature. So we'll try and stay at 37 degrees centigrade at all times. pH, however, changes drastically throughout the body. The mouth is about pH 7, and it drops down, as I said, to pH 2 roughly in the stomach, and then raises back up to slightly alkali in the intestines at pH 7.5. So the enzymes in the small intestine are slightly favored towards alkali conditions. I mean, they're virtually neutral, but very slightly towards the alkali conditions. And to help counteract the stomach acid of pH 2, because that leaves the stomach through the sphincter at the base of it into the intestines, the body mixes it with bile, which helps to counteract the stomach acid. Bile is quite a viscous substance and is bright green, actually, so as you can see in the vials there. And it helps neutralize the stomach acid. The problem with having such a low pH in the stomach is it actually is a very hostile environment and very dangerous for the cells there, because acid will effectively damage the lining of the stomach. So this can happen and we can end up with ulcers forming. Ulcers are formed where the acid has been exposed to the lining of the stomach. We've got a mucus layer there that helps to protect the lining of the stomach. But if that is thin due to illness or malnutrition or something like that, then the acid can actually burn the lining of the stomach. And that can cause a sore, which can become very painful. So that mucus lining is protective and prevents us from getting stomach ulcers, but can effectively be worn down due to different circumstances. And then the acids and actually the proteins within the stomach juices can work to break down and attack the lining of the stomach. As I said, bile, which is produced in the liver and stored in the gallbladder, will help to neutralize any acid leaving the stomach. As you can see, we've got the exit to the stomach here, so the stomach would be around about here. And as the contents of the stomach leave, they pass the ducts to the pancreas and the bile duct, which is leading to the gallbladder. So the liver makes the bile, it's stored in the gallbladder, and as the fluid called chyme leaves the stomach, it's mixed with the bile in this location here, and that helps to neutralize the stomach acid, stops us having any damage to the lining of the intestines. So, as I said, it's stored in the gallbladder, and any further acid will be neutralized there. So the enzymes that come out of the pancreas are designed to favor that alkali environment. Also, bile has another effect. It's not an enzyme, very important we understand that, but it does help to emulsify fat. What emulsify means, as it says there, is to break down big droplets into smaller ones. So it takes those big fat molecules, which are actually quite difficult to digest, and it breaks them down into tiny, tiny little molecules, which provide a lot more surface area for the lipase to work on. So you're exposing a lot more of that big fat molecule, no pun intended, into a lot smaller molecules, which then means it can be worked on by the lipase. Due to changes in diet, high protein diet, stuff like that, we can actually end up getting gallstones. These can cause a blockage in the bile duct, because what happens is the bile there can actually solidify down and create quite large stones, 2.5 centimeters across. And if that becomes blocked, it really disrupts digestion. So digestion can't occur because the enzymes are now being exposed to a more acidic environment, because the bile can't get to the intestine to neutralize it. So therefore the enzymes won't work as well. And also they're incredibly painful. They can cause swelling, severe stomach cramps, these sort of things. And nice little factoid to finish off on, you've probably heard of biological washing powder. As you can see here, this has got lipase in it and protease to help dissolve grease and dissolve protein. So biological detergents are used in the home quite a bit. We are actually using enzymes to help us clean things. What happens is the enzymes will allow us to work on the stains at a lower temperature to break down grease, break down bits of protein, that sort of thing that gets stuck to the clothes, as happens. And this will allow us to work at a lower temperature, which removes the stain, therefore using less energy and, really importantly, saving us money. Thank you. Bye.